Hello students, welcome to LEELA'S tutorial. So in last class we have discussed about protoplast isolation and culture. Right? What do you mean by protoplast? Protoplast is a plant cell without cell wall. Right? So here we are going to isolate the protoplast and then fuse the protoplast in order to produce hybrids and hybrids. Right? So let us see production of hybrids in detail. So here the development of somatic hybrids is also referred as somatic hybridization process, right? So in this process the name itself implies that the somatic cells or the somatic protoplasts are fused in order to produce a hybrid cell. So what happens when two protoplasts are getting fused, their cytoplasm and the nuclei will get fused in order to produce a hybrid. That means both the protoplast characteristics will be present in that hybrid one. So let us see in detail. So somatic hybridization process is an in vitro fusion of protoplast in order to produce a hybrid protoplast cells. When these are cultured in plant tissue culture media, they result in production of hybrid plants, right? So now these hybrid plants, why you are going to produce a hybrid plant when we have the natural habitat? Because we want a single plant to have more beneficial characters. So what we do, we select two different plants and then that means the plants belonging to the different species, not to the same species or the same plant, right? You are going to select two different plants which belong to the different plant species or genera, right? And then you are going to fuse the protoplast of both the plants so that you get both the beneficial characters in that hybrid plantlet, right? So here that is the reason in case of natural pollination or fertilization process or natural reproduction process, what happens? The same genome which is present in the parent will be inherited to the progeny, right? But here you want the progeny to have some other beneficial characters also, right? So let us see that. So first step is selection of two different plant species. So it can be a plant belonging to different species or plant belonging to different genera, right? So why I am specifically saying that plant belonging to the different species or genera? Because the natural pollination or the plants are sexually incompatible. Though the pollination occurs, they fail to undergo the embryo development, right? So that is against the nature, right? So here the selection of two different plant species or two different plants belonging to different genera can be selected because a natural process will not undergo. Okay, but we want both the beneficial characters. So for that reason we are going to produce the hybrid plants. Okay, so first it is selection of two different plant species. You are going to select, if for example if you are taking a flowering plants, right, whose flower is much more bigger in size in one species and in other species the color of the flower may be good, right? So you want both the characters to be present in that particular flower, right, in particular hybrid plant. In that case you are going to select one of the plant with the beneficial character and another plant with another beneficial character. So once you select the plant, you are going to take the leaves, because here we are going to take the somatic cells, right? Here the gametic cells are not at all involved. We are producing the plants by using the somatic cells. So first you are going to select the plant and then you are going to take the leaves and peel off the upper and the lower epidermis. Then put it in the solution so that the cell wall will be denatured. Okay, the cellulase, pectinase or macrozyme enzymes are added so that the cell wall will be dissolved. Okay, so when the cell wall is dissolved, you will be left with the somatic cell without cell wall. That is nothing but your protoplast. So once you get the protoplast, it is the isolation of the protoplast. That means you are going to purify the protoplast. So now when you are going for the denaturation of the cell wall, what happens? There may be some degraded cell material, cell wall material or epidermis. So everything must be clarified and you want only the pure protoplast cells to be present. So what you do? You go for the centrifugation process so that you isolate only the protoplast. Right? So two different plant species protoplasts are purified and isolated. Then next is you are going to add both the plant species protoplast in a container. Right? Then you are going to add the fusogen, fusion causing agent, so that the protoplast will fuse and then they result in formation of hybrids. So in that fusion also there may be a plant species. This is A and this is B. You want A and B cell to be fused. If it is A and A, that is of no use. If it is B and B, even that is of no use. So here again you have to remove these and you want only the hybrid protoplast. So here again screening and selection of your hybrid protoplast cells will be done. Once it is done, you are going to culture your hybrid plants. Okay, so culture is done in plant tissue culture laboratory. Next coming to the applications. Here you have, why you have to perform this somatic hybridization. The first reason is if the propagation is not possible in case of sterile plants, right, whose gametes are sterile. Otherwise in case of asexual reproducing plants. Otherwise, the plants which are sexually incompatible, right? That means they are sexually incompatible means both the here I have given the first hybrid plant which is produced is tomato. So normally if you take the potato and the tomato plant, they are sexually incompatible. Though the pollination occurs, the fruit or the plantlet will not be produced, right? That is called as sexual incompatible. So in that species also you can produce the hybrid plants. Okay. Next one is manipulation of cellular genome. If you are taking a single protoplast, it is having its cytoplasmic or the mitochondrial DNA in the cytoplasm and the nuclear DNA in the nucleus. If I am producing the plants using plant A, natural pollination, what happens? Only these characters will be inherited to the next generation. But here in this process, I am going to fuse the nucleus and also cytoplasm of two different plant species. So the progeny will have both the characters in their cytoplasm and also in the nucleus. Right? So here I am going to manipulate the cellular genome. Either, see, even here I can manipulate by introducing the gene of interest. Again that will come under your genetic engineering. Right? I can introduce some gene of interest, then I can have both the characters of two different plants. Right? So like that I can manipulate the cellular genome or modify its genome, or I can go for the plant breeding process. Right? So in plant breeding, we want some new or beneficial characters to be present in the crop plants or the agricultural plants. Right? The next one is to generate the novel phenotypes. So the new phenotypic character, the phenotype is the external morphology of the plants. Right? The mostly what we want, the external morphology, the color of the flower. Right, or the size of the flower or size of the fruit or color of the fruit, which is seen externally. So that can be generated. The plants with novel phenotypes can be generated. Then next one is the desirable variations in the plants can be obtained. So here the desirable variations means here you can see if you want the plant to produce some other beneficial character, even that variation by chromosomal variations, you can again modify the genome or the chromosomes. Right? Then next one is production of intergeneric and interspecific hybrids. As I told, the tomato, right? So here as they are sexually incompatible, I can produce a hybrid with both the characters. Right? So that is both the characters, or I can say I can produce a hybrid plant in between two different plants of different genera or two different plants of two different plant species. Right? So these are the applications. If you see diagrammatic representation here, the tomato is the first hybrid plant which was successfully produced. So here they have isolated the protoplast of tomato and potato. Suppose if it is a protoplast A and protoplast B, by means of the fusion causing agent, that is fusogen, that is PEG, PEG, polyethyl glycol is the fusogen used here. That means this is the chemical method of fusion, protoplast fusion. In this case, what is happening? Both the protoplasts come closer. So here you can see the protoplast fusion process, where gradually the membrane, the plasma membrane present in between two protoplasts got dissolved, right? And first it will result in fusion of cytoplasm, followed by fusion of two nuclei. This is called as heterokaryon stage, where you can see the single cell consisting of two nucleus, right? The two nucleus of two plant protoplast cells are present. After the fusion of cytoplasm and the nucleus, you can see development of hybrid protoplast cell. So now these hybrid protoplast cells are inoculated in the liquid media and they are cultured to form the callus. Once the callus is generated, again by varying the auxin to cytokinin ratio, you can allow them to develop into an embryos, right? So again the embryos, they have different stages such as globular, heart shape and torpedo shape, which gradually transforms into an complete plantlet by producing the root and the shoot system, right? So here by like this you are going to produce a hybrid plantlet using your hybrid protoplast culture in plant tissue culture, right? If you have any doubts, you can put it in comments.